is only really secreted when you eat greasy food. You have amylase, which you generally speaking, once again, only find in the saliva when you eat food high in carbohydrates, right? Mucus. The mucus is really there to help to form the bolus when you're swallowing. So we can say that these three here, generally speaking, these three components of saliva are more directly related to when you're actually trying to chew something, right? You're eating something, as we say. Alkaline electrolyte solution, that's more general. The reason it's more general is because, as I mentioned to you before, the pH of your mouth is supposed to be kept somewhere around 78, so it should be slightly alkaline. This, of course, is to reduce the impact of bacterial acids. So the alkaline electrolyte solution in your saliva helps to keep your general mouth fluids slightly alkaline, right? It also mentions here that saliva will contain thiocyanate and lysozymes. Now, these are designed to destroy bacterial cell walls. Okay. Also, you can have antibodies also being released into your oral cavity to help control bacteria as well, right? So saliva has a lot going on for it, right? This is the reason why I mentioned to you that if you happen to be somebody who is cursed with dry mouth, and you do have some people who literally do not produce enough saliva, it's a chronic condition. There's not much you can really do about it. The problem with chronic dry mouth is because you don't have the saliva, or you produce it in smaller concentrations than normal. These people have a hard time swallowing. These are the people who tend to swallow and I hear them choking and coughing because they can't get enough saliva worked up to form a proper bolus, right? So they won't be able to get like the mucus, etc., in the saliva to form that bolus. But people with chronic dry mouth also tend to have bad breath, right? I don't mean like morning breath or you just eat something nasty breath. I mean, even if they brush their teeth, you check them an hour later and their breath smell bad. Or not too long after they eat something, their breath start to smell bad because of the bacteria breeding up, simply because they will not have been able to produce enough saliva to coat their teeth and gums with the saliva, which would of course contain the lysozymes and the thiocyanates and the alkaline electrolyte solution to help control the bacteria, right? Anyway, um, how do you control secretion? So, no surprise, peripheral nervous system is going to increase salivation. Remember, as we said before, in the digestive system, the peripheral nervous system speeds up activity, right? So the SNS, of course, is going to slow it down. So the sympathetic nervous system slows things down, right? So small amount of saliva or thick saliva. So if you they, y'all did fight or flight last semester when with Mrs. Longman, right? So they would have told you about some of the things that happen during fight or flight. When you get afraid, you get afraid of something and you feel like your mouth dry out, right? You feel, as I say, you feel like you have ashes in the mouth. That is because, of course, of the sympathetic nervous system activation, right? Now, why do you want to drop saliva? Imagine you're trying to run, right? Something chasing you. You're breathing hard, you're breathing through your mouth because you're running hard. You need to get as much oxygen in as possible. And your mouth is full of saliva. You're going to inhale that saliva into your lungs. The last thing you want to be doing is choking on spit when you're trying to run for your life. So, of course, if everything else, if the sympathetic nervous system is activated, you have to dry out the mouth just in case, right? Now, it mentions here you have other brain nuclei and higher brain centers that can also impact salivary secretion. So, chewing gum. As you're chewing something, technically it doesn't have to be gum, because you see the same thing when you have people who chew plastic. I remember back in the good old days when we had plastic straws, you chew the plastic straw, just like you're just sitting back relaxing after you eat your food. You're just chewing on the straw. And you'll be making saliva as you're doing it. And funny enough, as you were chewing on the straw after you ate your fast food or whatever it is, it would actually help to clean your teeth, right? Because the plastic straw was insoluble and it would rub against your teeth and your gums. And in addition to that, you'd have been producing extra saliva. But of course, these days we don't have those advantages because apparently we're killing turtles or some nonsense. Now, smelling food and thinking about food can also activate saliva production. Think about it. For most of the life of our species, if you are close enough to smell food, it means you're getting some of it, right? Therefore, smelling the food and starting to salivate means you're preparing your body to receive the food. So you're preparing your mouth to receive that saliva. Um, that extra saliva is there to help to receive the food and to make the bolus and all of these things, right? Now, thinking about food, same deal, right? Then, of course, it mentions irritating stimuli in the esophagus. Maybe I know maybe this has happened to you before. You smell something, or and then your mouth start to water, it spit, and people spit out the saliva, and you say, boy, you smell something that, for example, you maybe walk past some lovely fresh dog doodle, and get that lovely whiffing, and all of a sudden your mouth start to salivate. Obviously, not because you want to do it. At least I hope not, right? But it is actually a response to a disgusting smell, right? So yeah, there are various things that can actually activate salivation, right? I'm not gonna spend very much time on the teeth. Um, I will simply say this slide here charts, generally speaking, the type and number of teeth. Give this one a look. We're not going to spend very much time on teeth, right? Just be aware, of course, of the function of teeth. And I don't think anybody in here doesn't know what your teeth are for, right? At least I hope not. Now, pharynx. Thankfully, we did pharynx when we were doing respiratory system. So we all know that you have nasopharynx up near the nasal canal. You have oropharynx behind the tongue, and laryngopharynx, which is supposed to be the common point for air and food to pass, right? So all of that should sound familiar to you already. Esophagus, we already know esophagus is just that tube that runs from the bottom of the laryngopharynx to the stomach, right? Just that length right there. It mentions here is a hollow muscular tube that transfers solid food and liquids to the stomach. That's all it's for. Uh, Sorry, and it extends from the cricoid cartilage along the posterior surface of the trachea. What is it saying? Remember, cricoid cartilage is the lowest cartilage. So if you look behind the trachea, behind the larynx, right down at the very last cartilage, that is where the esophagus opens up, right? And it runs from there all the way down to the stomach. Now, you have several sphincters associated with your stomach. The cardiac sphincter is going to be the first one you come into contact with, also known as the esophageal sphincter. It is going to essentially prevent substances from your stomach from leaving and going into the esophagus, right? Now, I'll speak about the importance of the sphincter in a minute. Now, it mentions here that, as you know, because we did it in lab, at least some of us have, the mucosa of the esophagus is stratified squamous epithelium. And the reason for this is very simple. As you're swallowing the partially chewed up food, there still might be sharp, hard bits in there, which will be rubbing against the inside of the esophagus. Same thing with the inside of the mouth. Same thing with the back of the throat, the oropharynx and the laryngopharynx. So because of that, you need to have stratified squamous tissue here to resist that abrasion, okay? Now, Going down now, the submucosa, remember the submucosa is below the mucosa. That's why it's sub. Um, you're going to have mucus secreting glands, lots of them. Now, why do you need plenty of mucus secreting glands? Because you're trying to move a bolus of food along, it needs to slide down the esophagus. So by coating the esophagus in mucus, of course, it makes it easier for your bolus to slide down. So that should be fairly obvious, right? You also, below the submucosa, have your muscularis externa. Muscularis externa, smooth muscle, two types. You have the inner circular muscle and the outer longitudinal muscle. Now, that is the bulk of it, but as it mentions here, the first third of it actually contains skeletal muscle, right? Now, why do you want skeletal muscle in the first third of the esophagus? Skeletal muscle is voluntary muscle, right? What this means is if you start swallowing something, if you change your mind fast enough, you can actually squeeze it back up. Um, Now obviously this is not something you do on a regular basis, but have you ever seen one of these acts where you have like a magician or something, they'll take like a rubber ball and they'll put it in their mouth and they'll close their mouth and open it and there's no ball in the mouth. Now you might say sleight of hand and they are hiding the ball, and in some cases this is. But you do have some people who actually swallow the ball down, hold it in their esophagus because they've practiced, and then they can bring it back up whenever they feel like. Right? So that actually does happen, right? As a matter of fact, it's something that guys do in prison, believe it or not, where they'll know they're going to prison. So they want to smuggle in some drugs. So what they'll do is they'll get a condom and they'll fill it with the drug of their choice. They will tie a string around the condom. Then they will tie the string around the back, one of the back teeth, and they'll swallow the condom. The condom, of course, on the string is now hanging in the esophagus, right? In the upper third of the esophagus. So assuming they get past the guards and go in, they can reach back and start tugging on the string. And as they tug on the string, they can actually have to squeeze the condom back up out of their esophagus as well, right? That's one of the reasons why in prison movies, if you've ever seen a prison movie, when they're taking in the inmates, they tell them to open their mouths because they're looking for strings hanging up things around the back, right? All right. That said, the middle third is a mixture of skeletal and smooth muscle. Remember, circular, longitudinal. And then now, by the time you get to the lower third of it, that is all smooth muscle. So once you get to the lower portions of the esophagus, it is all involuntary from that point on, right? Now, I should also mention sword swallowers. This is also the reason why people can swallow swords, because they're sliding the sword down. They're able to relax first half, the first two-thirds of the esophagus, which makes it easier for the sword to slide through, right? And then they can pull it back out. Um, Still very dangerous because I know of one person who swallowed swords and he sneezed in the middle of it and the sword cut him open. So yeah, you can still die. Anyway, I also mentioned in class, in the labs, that there is no serosa around the esophagus. Instead, you have an adventita. The adventita is designed to connect the esophagus to the surrounding tissues in the neck and the upper part of the chest. All right. Remember in the rest of the digestive system, well, most of the rest of the digestive system, you're going to have the mesenteries, which help to hang your tubes. There are no mesenteries for the esophagus, so it needs to be attached in another way, hence adventita, right? Now, obviously, we are doing drawings of the digestive tract in the lab this week, if you haven't done them yet. So this diagram should already look very familiar to you. The parts you see listed here are the ones we're expecting to see in your esophageal drawing, right? You're doing a section of the esophagus. But in that section, we are expecting to see the stratified squamous layer of the epithelium right here. Then below that, the lamina propria. And then now below lamina propria, you're going to have a layer of muscularis mucosa. And those three are going to be the mucosa. So one more time. The epithelial layer, stratified squamous, lamina propria, muscularis mucosa. Those are all parts of the mucosal layer. Okay? Then the submucosa, of course, will have glands, etc., embedded in it. Then below that now, you're going to have your muscularis externa. For the most part, for at least for our drawings, we're looking at two sections because normally our section is from the lower esophagus. So you're going to find circular muscle first, then in between the circular muscle and the outer longitudinal muscle. So here we have the circular muscle, and this is the longitudinal muscle. In between, which is this area right here, as it mentions here, this is the myenteric plexus. Okay, and this is going to be important later on because, as you know, your your digestive tract is running a separate subsection of autonomic nervous system, which does not require direct input from your brain. And the myenteric plexus is a part of that, okay? Now, of course, the outermost layer is going to be adventita. And I've already explained to you why it's adventita and not serosa, okay? So, cardiac sphincter, we're come to it. Now, it is a funnily named sphincter, if you, also known as the lower esophageal sphincter. Why is it a cardiac sphincter? When you hear cardiac, the first thing that comes to mind, of course, is the heart. So why is it a cardiac sphincter? It's because of heartburn. Heartburn is caused when you have a mixture of stomach acid normally and food that gets past your lower esophageal sphincter, your cardiac sphincter. It may be you're belching or something. And as the sphincter opens to allow the gas to pass out, some of the fluid or acid chine gets pushed up the esophagus and coats normally the lower esophagus. And of course, because it's so acidic, it starts burning the tissues of the lower esophagus. And the lower esophagus runs pretty close to the height of the heart. All right? So therefore, heart burn. The esophagus runs posterior to the heart, and the part that is burning is right in line with the heart. So you feel like you feel like it's burning through your chest, hence heartburn, right? But obviously it has nothing to do with the heart. All right, that said, let's talk about the swallowing. What steps are required for you to swallow something? So there are three phases involved in swallowing. Some of them are voluntary, of course, and some are involuntary. So the oral phase, pharyngeal phase, and esophageal phase. Now start thinking which ones are voluntary and which ones are involuntary, right? Now, it should come as no surprise to you that the oral phase is voluntary. You choose to put things in your mouth. You choose to chew them. You choose to swallow them, right? It mentions here that in order to swallow in the oral phase, the first thing you have to do is form a bolus. So you have to take the food in and chew the food up nicely, mix it with that saliva to form this mushy mess. And then if you'll notice here, here's the food, tongue pushes up and back. You're literally shoving the food backwards using the tongue, back towards the oropharynx, which of course, lower part of the oropharynx, as you mentioned, that is where you're going to have the opening to the esophagus, right? All right. And if you notice, while you're swallowing, The soft palate, which is right here, it raises up, okay, to get out of the way. So the soft palate goes this way. That's why it ends up like this, okay? Which of course opens the opening towards the back, making it easier to push your bolus towards the back. Pharyngeal phase, of course. Now the bolus is now in the oropharynx and sliding down to your laryngopharynx. It mentions here that you have tactile receptors on the palatal arches and on the uvula itself that are stimulated by the bolus. So the bolus is going to press against these receptors and this triggers the swallowing center in the medulla oblongata, and this causes the pharyngeal muscles to contract, right? So basically, if you this is maybe you've seen movies where somebody's unconscious and they need to swallow something, and when you do, you push it towards the back of the person's throat, and sometimes they'll just automatically swallow. Or in some movies, you'll see they put it in the back of the throat and then they'll rub the throat, trying to stimulate the swallowing response. What they're basically trying to do is put pressure on these receptors here and cause them to force you to swallow automatically, right? Now, as you're swallowing, it mentions here that the larynx is elevated. I'm assuming it makes sense to you that if you elevate the larynx, right, and you close off the larynx using your epiglottis here, you're going to force your bolus to go down the esophagus and not enter the larynx, right? So the larynx goes up a little bit, then you slam the epiglottis closed, food then can and water can slide past, no problem, right? And while you're swallowing, of course, because you don't want to risk food getting in to the larynx, the respiratory center is going to inhibit breathing. Even if you have nothing in your mouth but saliva, you can try it yourself. Try to swallow and take a breath in at the same time and see if you can manage it. So let's just start breathing in and then and then as you're breathing in, try to swallow. You're going to find it's impossible for you to do. Okay? This, of course, is a protective mechanism. I think I already mentioned to you when we did respiratory system that babies can, however, breathe and swallow because the milk is trickling, because you lay the baby basically on the inclined on the back, which allows the milk to trickle down the back of the throat and into the open esophagus. So, and because of that, the probability of it getting into the trachea is lower. So babies can breathe and swallow, right? Anyway, the esophageal phase now. So once the bolus moves now into the esophagus, it mentions here that you're going to start off peristalsis. You all know from C-sec already, peristalsis, muscular waves of contraction that are designed to push the bolus from the mouth to the anus as they would have sold at C-sec. But basically, all we're saying is that you're moving the bolus in one direction only, right? That's the norm. So you're forcing food down the esophagus into the stomach and then from there on to the rest of the digestive tract, right? So of course the bolus gets pushed all the way down via peristalsis to your lower esophageal sphincter or your cardiac sphincter. And the pressure of the bolus on the sphincter causes the sphincter to open and the bolus can then slide in. And of course the sphincter closes behind it, okay? Now, the closed sphincter is important because it not only does it close, but it closes strongly. Now, because of that, your movement, your three-dimensional movements in the environment will not cause the sphincter to open and anything to dribble out of the stomach. We already spoke about why things leaving the stomach are bad, because you will end up with something, as we mentioned, to like acid reflux, heartburn, etc., right? So you can stand on your head with food in your stomach. The esophagus, the lower esophageal sphincter or cardiac sphincter will stay closed, right? As you're jumping and skipping and hopping and doing all of these other things, it stays closed. Now, obviously, if you overload your stomach and then you try to lie down, then the force of the food pressing against the sphincter can cause it to pop open. So if you eat in excess and lay down, you can end up vomiting all over yourself, right? All right, stomach now. Now, obviously, one of the main functions of the stomach is storing the food that you've eaten, okay? Now, the average person can store 2 liters worth of food in the stomach at any given time. 2 liters might not sound like a lot, but 2 liters of meat, that's basically 2 kilograms. That's essentially 5 pounds of meat. That's not bad, right? So it mentions here, in addition to storage, the stomach is involved in mechanical breakdown of ingested food. Now remember, chemical digestion is a thing. Mechanical digestion, we don't say that anymore. We say mechanical processing because you're taking big food particles and you're crushing them down to make small food particles. You're not breaking down molecules, hence mechanical processing, right? Or breakdown in this case. It also mentions here some chemical digestion will occur because you can see you're disrupting chemical bonds in the food. Once you take big molecules, as I'm sure everybody knows, and turn them into smaller molecules, you're doing actual chemical digestion, right? This, of course, is going to be mainly for proteins, but we'll talk about the lipids in a minute. But in addition to this, your stomach is also involved in absorption of vitamin B12. Okay? Now, I don't know if any of you know what B12 is for, because there are a whole range of B vitamins, right? Um, but B12 is, is important for combating anemia. It is also important, particularly for pregnant women, because there's a direct impact on the development of the fetus, fetal brains as well, right? So yeah, the ability to absorb B12 is pretty important for your blood, pretty important for the development of a fetus, right? Then now, let's look at the stomach's gross anatomy. You're going to notice here that the stomach has 2 curves. You have a greater curvature and a lesser curvature, which is similar to what we mentioned, well, which what I would have mentioned, hopefully in the video about urinary system. I would have mentioned that you have a greater curvature, lateral curvature, and a medial curvature on this in the kidney. Here, you once again have a greater outer curvature and a lesser inner curvature, right? Now, in addition to those 2 nice big curves, you'll notice that the upper part of the stomach is referred to as the fundus. You can see the esophagus comes in, and the area where the esophagus attaches to the stomach is called the cardia, because of course, heartburn, right? And then the fundus rises up from the cardia. Now, below the cardia, you're going to have the body, which is the bulk of the stomach. And then down here and down towards the end of the lesser curvature, as you can see here, where the stomach is narrowing, you're going to get the pylorus. All right? The pylorus, of course, has sections. I mean, most of these things have sections. I'll talk about the sections in a second, right? So remember now, we have a fundus, which is the highest point. We have the cardia, right beside where the esophagus meets the stomach. We have the body, which is the bulk of the stomach. We have the pylorus, which is the region where the stomach starts to narrow. Okay? All right. Now, in terms of the muscles associated with the stomach, I've said it multiple times, but let me say it again. I said it multiple times yesterday, but let me say it again today. you're looking at three layers of muscle. Okay? You have the oblique muscle. It is the very first layer of muscle. I mean, it's not a very, very thick layer of muscle, but it's the first one. I want you to notice something here about the muscle fibers, right? Because this one actually shows, this diagram shows the muscle fibers. I want you to notice here that the muscle fibers are running sort of like this. Okay? They are running diagonally, basically. Okay? Then I want you to notice the circular muscles, which are running more horizontally. And you can see here from the longitudinal muscles, they look like they're starting to run somewhat more vertically. As it leaves, as you can see here, as it leaves the cardia and comes down along the greater curvature, it's like the muscles are curving, right? So we're basically looking at horizontal, vertical, diagonal. Okay? So once again, diagonal for the oblique, more horizontal for the circular, and more vertical for the longitudinal. Now, why is this important? It means, of course, that these three groups of muscle can contract and pull the stomach in three different directions. You can squeeze the stomach in diagonally, they can squeeze it in horizontally, and they can squeeze it in vertically. So why are you squeezing in all of these different directions? It allows you to mix the content of the stomach more efficiently with the gastric juice. Okay? And gastric juice. Now, there are lots of juices in the digestive system. I've always found the idea of calling this stuff juice to be very off-putting as somebody who likes juice. But yeah, remember, stomach has plenty of juices. I wouldn't advise you want to drink any of them, but it is juice, right? So gastric juice, of course, is going to contain a cocktail of different substances, including mucus, because mucus is always there somewhere. You're also going to have hydrochloric acid. Everybody knows that one. You're also going to have some amount of water because, water is also always there. You're going to have pepsinogen or the active form pepsin, right? Um, Various other secretions, hormones, etc. Some of them will actually be in the stomach itself. So all of these will be within the gastric juice. And you'll be mixing your food with this juice and be able to churn three-dimensionally. Helps you to mix more efficiently, right? Okay. Now, I also want you to notice that within the stomach, the lining of the stomach, you have these weird valleys, right? These are your rugae. Now, the rugae look very similar to the trabecular carni in the wall of the ventricles. If you remember that. But these rugae here actually allow the stomach to stretch, to distend, as we say. So as you're eating more and more food, the rugae actually smooth out because of the pressure of the food, allowing the stomach to expand. So in a very real way, the rugae are sort of like elastic bands or a sheet of elastic. Now, as you go from the rugae, I want you to notice we're going this way now. So this is the body. We're moving from the body where we're doing all that mixing and churning. What we have down here now, after you do all the mixing and churning, what we have here is chyme. So when you've thoroughly mixed the food that you've eaten and swallowed with the gastric juice, you get this mush called chyme. The consistency of the chyme can vary from being extremely liquid to being semi-solid, right? And what happens now is that you're moving the chyme from the body through the pylorus. First part of the pylorus here is the antrum. So you're moving the chyme from the body to the antrum. And as you do that, You then move to the pyloric canal, which as you can see here is this very narrow section right here. And this narrow section moves right up to the pyloric sphincter, which is the next sphincter associated now with your stomach. Okay? Now, the pyloric sphincter controls the movement of substances, no surprise, out of the stomach. Normally when you say control movement, we say in and out, right? But you really should not be sending anything in the opposite direction. It really should only be going in one direction. Though in extreme cases, you can actually have bile being pushed up through the pyloric sphincter back into the stomach, and then people will vomit it out. All right? So, yeah. So the pyloric sphincter opens up to allow this acid chyme to leave the stomach and enter the next tube, which of course, as I'm sure you all know, is going to be your small intestine, right? And the first part of the small intestine, of course, is the duodenum, or as my lecturer used to say, the duodenum. However you prefer it, it's fine with me. So remember the parts: cardia, fundus, body, and of course the pylorus, okay? And, and of course the curves are easy to remember, right? All right. Now, let's give you a little bit of detail about what's happening in these particular regions. Now, the cardia and near, this is lower esophageal sphincter. That area there is going to be high in bicarbonate containing mucus, right? So the area, so that the wall right there is just lined with very thick, very alkaline mucus. And of course, this is to prevent acid reflux. It's to stop food from leaving the stomach and going back into the esophagus. As I mentioned already, the esophagus is not designed to deal with acid chyme. So the LES is supposed to stop that from happening. And of course, if it does happen, you're supposed to have this bicarbonate-laden mucus to help to at least minimize the acidity of the chyme leaving, because you have to push the vomit into the mucus as you push it out, right? Then now the fundus and the body. You'll notice here you're going to find protons, right? So H. You'll find the intrinsic factor for the absorption of the B12. You'll find bicarbonate-rich mucus again, lining the walls, because guess what? The protons, H plus, as we know, that's what actually makes things acidic. And the wall of the stomach is made of proteins, right? It's made of cells, just phospholipids and proteins, which means, of course, that an acid can eat through it too. So your stomach is not immune to acid. It's just coated in this very rich bicarbonate mucus that reduces the impact of the protons on the walls of the stomach. And I say reduce, not stop, because your stomach lining is constantly being regenerated because the acid in your stomach is eating it away, right? And of course, we also have pepsinogens. Pepsinogens, of course, will form, as I'm sure you all know, pepsin. And everybody knows from CSEC that pepsin is there to convert proteins to polypeptides, right? It helps to break down proteins. And where's a good place to find proteins? The stomach wall, right? So that's another thing the mucus helps to prevent from reaching the wall of the stomach. It helps to prevent pepsin from getting there and also eating through it, right? Now, lipase. Babies produce something called gastric lipase. Gastric lipase is not really something you find very much in adults. It's not in any significant quantities. So why do babies need gastric lipase? Remember what a baby is feeding on, okay? Baby's feeding on milk. And that milk is going to be very high in lipids, lots of fatty acids in there. So if you want to break those down, the gastric lipase is important. And to be honest, the strength of the stomach acid of our infant is not that great, right? I mean, remember what we need the acid for. We need the acid to activate the pepsinogen to form pepsin. Okay, so we have to have that HCl here, and HCl is also used to control bacteria. Right? It cleanses the chyme. Now, if the infant is getting mother's milk, it's less likely to be swallowing bacteria. So it doesn't need extremely strong stomach acid, right? And because of that, it can have gastric lipase, which can still function in the acidic environments of the baby's stomach same way, right? But of course, as you get older, start eating solid food from random places, or if you've ever been around toddlers, shoving random things into their mouths, you know, of course, at that stage, you need to start strengthening the stomach acid because if you take your eye off of them, the child will commit suicide, you know, just randomly. So you have to be very careful with them, right? Anyway, now it also mentions here that the fundus is a reservoir of a tonic force during emptying. So when you're vomiting, the fundus in the body will contract to push out the food that you're trying to vomit out, right? Then on the lower part, antrum and pylorus, once again lined with lots of bicarbonate mucus. So pretty much you find bicarbonate mucus in every single part of the stomach, right? And the antrum is quite involved in the final mixing and grinding of the food in the chyme before it passes it to your duodenum, right? And of course, here are the rugae in the stomach. Now, as I mentioned in class yesterday, the epithelial lining of your stomach is going to be simple column epithelium. And the simple column epithelium is also going to contain goblet cells, right? As you know from ALP1, goblet cells produce mucus. As I've just been saying to you, if there's one thing you need a lot of, it's mucus, right? So the gastric pits open onto the gastric surface. So basically, you have the epithelium forming what we call gastric pits, and you drew the stomach epithelial yesterday, and so you remember. Hopefully, you remember that there are pits there. And glands now are associated with these gastric pits. So the idea here is that epithelium forms these pits, these pores, and lining the pores, you have cells that are going to be producing various substances and dumping them into the pores, right? So this, of course, is a section through your stomach. This one here, I would say, is important. So here you can see the upper mucosa of your stomach. Here you have the gastric tissue. So you can see the pits here. And within the pits now, as I mentioned to you, you're going to have various types of cells. And the cells are going to be extremely important, okay? Here we have a submucosa. And the submucosa, as you can see here, contains various types of blood vessels and lymphatic vessels. Also, nerves are associated with it as well, right? Then you have muscularis externa below. Remember, all sections of the digestive tract have the same layering, as I mentioned before, right? So, mucosa, submucosa, muscularis externa, all have those. Esophagus has adventita, but the stomach has a serosa. Because why? Because the stomach is surrounded by the mesenteries, right? And of course, we have our oblique muscle, circular and longitudinal muscles there, right? This one should be an easy one for you to remember. So this is just showing an electron micrograph of the actual openings to the gastric pits, right? Now, this is another slide I think is pretty important in terms of the information that's on it. You'll notice here that as the epithelium dips down to form the gastric pit, the upper part of it, we call it the neck. Okay, so that's the neck. So if you notice here that near the base of the neck, you have mucous neck cells. And of course, no surprise, it produces bicarbonate-rich mucus to help protect the lining of the stomach, as well as to mix with the food in the stomach as well, right? Then below the mucous neck cells, you have parietal cells. And the parietal cells are associated with hydrochloric acid production. Then they also are associated with the release of intrinsic factor for vitamin B12 absorption, right? Then we have enterochromaffin-like cells, and they produce histamine. Now, you know histamine causes swelling. So what do you do with histamine down there? When the histamine is released, it does cause swelling as well, and the swelling squeezes down on parietal cells and stimulates them into releasing hydrochloric acid. Then we have my favorite type of cell here, a chief cell. And the chief cell produces pepsinogen, not pepsin, okay? Chief cells produce pepsinogen. Pepsinogen is, as I'm sure you all know, the inactive form of pepsin. Pepsin has to be secreted in its inactive form, or it will eat through the stomach wall, as you know why. Enzyme dissolves protein, destroys proteins. Stomach is mostly protein and phospholipids, right? It also will cause gastric lipase to be produced as well and released. Then below that, now you have your D cells, which produce somatostatin. And somatostatin, of course, actually will reduce the influence or the impact of things like your enterochromatin-like cells, because it is trying to inhibit the production of acid. Then you have your G cells, and your G cells produce gastrin, and the gastrin will stimulate the release of stomach acid as well, okay? So D cells release somatostatin, which will inhibit the production of acid. Enterochromatin-like cells will stimulate acid release. G cells stimulate acid release, okay? So two are stimulating, one is inhibiting, right? So this is now showing you how you get the hydrochloric acid. So here we have our parietal cells in our gastric pits. Water plus carbon dioxide gives us our carbonic acid. Now this should sound familiar to you already, because what I'm describing to you is what happens in the transport of carbon dioxide, right, in the respiratory system, right? But notice here now, when you have the bicarbonate uh, breakdown, so now you have your bicarbonate ion and your proton. Remember, in the respiratory system, the proton gets attached to hemoglobin and it's carried to the lungs, whereas the bicarbonate ion just stays in the blood and gets carried in the lungs, right? But nowhere in the stomach, so obviously that's not going to happen. In the stomach, the bicarbonate ion is passed to the blood leaving the stomach, right? Now remember, you're producing acid because you've eaten something. So this is happening during a meal, after a meal, right? Or just before you started eating. That now means that blood leaving the stomach is going to become more alkaline because you're loading it with bicarbonate ions. That causes an alkaline tide. Okay? So if you check the pH of the blood leaving the stomach, it would come back alkaline. And this, of course, is a temporary form of metabolic alkalosis. So simply put, After you've eaten a heavy meal, your blood becomes more alkaline because of this alkaline tide. Now, while that's happening, the protons that we've made as well are being pumped into the lumen of the stomach. And while you're pumping the protons out, you're pumping potassium in. Okay? So this is an anti-port system. And in co-transport, you carry one thing, then the other one comes along with it. In anti-port, you carry one thing in one direction, and the other thing goes in the opposite direction that you're carrying things to, right? Simple as that. So if you're moving protons into the lumen in the antiport system, the potassium has to move out of the lumen, right? And eventually make its way to the blood. So now we have the protons, okay? But where do we get the chloride ions from? You get the chloride ions, of course, from sodium chloride, salt. We use the sodium for some stuff, as you know, plenty of stuff. The chloride ions can then move from the blood through the stomach wall and then be transported into the stomach. You can then combine, of course, proton, chloride, you now have your stomach acid, your hydrochloric acid, right? Pretty straightforward, I hope, right? Now, as we mentioned before, hydrochloric acid is going to kill bacteria. Okay, that's one of its major functions. It denatures proteins as well. Hopefully, you remember what denaturing entails. Y'all already know that proteins have different structures, primary, secondary, tertiary, quaternary structure, right? And depending on how the proteins are coiled up, it can be very difficult for enzymes to get down into this massive coiled structure to break it down. So basically what you do when you're denaturing the protein is that you're breaking the bonds holding the protein coiled up, and you're forcing the protein to stretch out. And as the protein stretches out, it's easier to cut it into pieces. Hence, you go from proteins to polypeptides. Okay? So that's what denaturing does. It aids in this process by breaking the bonds and stretching the protein out a bit. Now, it also mentions that hydrochloric acid can also break down plant cell walls as well as connective tissue in meat and provides an acidic environment, of course, for pepsinogen to pepsin reactions, right? We already mentioned what chief cells do, as it meant, but it does say here now that the pepsinogens converts the pepsin in a pH of 1.5 to 2. Now, surprisingly, you think the more acidic things are, the better for the enzyme to function. But here's the thing: the pepsinogen, if it gets too acidic, actually becomes denatured itself. So a pH that is lower than 1.5 actually starts damaging the pepsinogen. You actually get less pepsin being produced. So pH 1.5 to 2 is what you're looking for in the stomach, right? And we already mentioned that gastric lipase is produced in infants. I may not have mentioned renin yet. But we know in the renin-angiotensin system, I think I mentioned it there. But here's the thing. In infants, renin causes the coagulation of proteins. So the milk comes in and because lots of milk proteins in there. And you want to give the stomach acid, the gastric lipase, other things in the baby's stomach, time to work on the proteins and break them down. So what do? You produce renin. So by causing massive clots to form, or coagulates if you prefer to form, then you slow down the movement of the milk through the baby's stomach and moving into the small intestine, right? Giving time to properly break down this milk, to properly denature these proteins, etc., right? Once again, as you get older, of course, you no longer require the renin because you're not drinking that much milk. Okay. Now, Pylorus, of course, contains glands. No surprise, mucus secreting glands. Pretty much every part of the stomach has these, right? But you also find in the pylorus um, gastrin being released by G cells. And of course, this gastrin, as we mentioned before, stimulates the release of parietal and chief cell secretions. Okay? And this helps, of course, to promote also the mixing of the substance in the stomach. So basically, the gastrin helps release the hydrochloric acid, right? Helps to release the pepsinogen, and it also triggers the churning of the stomach, which helps to mix everything together so that you can break down your food more efficiently, right? Then your somatostatin now, by the D cells, as we mentioned before, will inhibit this release. So you can imagine now when you're eating food, let's say you're eating complex food. Pizza. Pizza is complex food if you think about it. Proteins, carbohydrates, lipids. If you're eating pizza with vegetables on there, you have your fibers and various vitamins, complex food. So the gastrin, of course, would become activated on a slice of pizza, and you stimulate the parietal and chief cells to do their thing. But let's say instead of you miss breakfast this morning, the last thing you want is stomach acid, because once again, the lining of your own stomach is in danger from the acid. So you can imagine the somatostatin being released to prevent the release of gastrin and therefore the stimulation of the parietal cells and chief cells, right? So how do we regulate the release of gastrin? It's controlled by the central nervous system in this case here, but it's regulated by short reflexes of the enteric nervous system in the wall of the stomach. Now, remember, the enteric nervous system is a specialized part of the autonomic system that is designed to keep the stomach functioning without input from the brain. Okay? You don't have to send impulses from the medulla to your digestive system for it to function. It can happily function without any instructions, right? So for example, this idea about short reflexes of the enteric nervous system in the wall. There are stretch receptors in the wall of the stomach. If you're eating food, of course, the weight of the food is going to stretch the stomach. This triggers the enteric nervous system, which can then cause the triggering of muscle contraction to help churn food. And that, of course, can also lead to the triggering of the release, once again, of gastrin to release substances from the parietal and chief cells, right? All right. Now, we're going to look now at the overall control over the gastric system, right? So it says here, regulation of gastric activity. This one here, well, let's run through and see how easy it is for you to absorb this one, right? When it comes to controlling the stomach, so we're talking about gastric control. Of course, that's stomach-related control, right? We're looking at three different phases. You have a cephalic phase. So I'm talking about the stomach. And the first thing I said to you is something to do with head. Because obviously cephalic means head, right? Then one of them now says gastric phase. And the last one is intestinal control. So what I'm telling you is that your stomach is controlled by your head, your intestines, and only partially by your stomach. Which sounds weird, right? But see if this makes sense. In the cephalic phase, you're triggering stomach activities based on what is being encountered in the environment. It mentions here you begin the cephalic phase before the food enters anything anywhere near your stomach. When you see food, when you smell food, when you think too much of food, when you taste food, your stomach starts to activate. It starts contracting and relaxing the muscles, right? It starts stretching out, awaiting the arrival of food. It's getting ready, it's priming itself. Gastric phase, of course, food actually enters the stomach, and the weight of the food further causes the stomach to relax. causes the release of the various gastric secretions in the gastric juice. So you can break down now whatever it is that you've eaten, right? Then now intestinal phase, you're looking at the presence of food now, or in this case, let's call it acid chyme. Because by this stage now, you've mixed the food with the gastric juice so thoroughly that you don't really have food anymore. You know, we'll just call it chyme. And because of the stomach acid, the chyme is acidic, so it's acid chyme. So that acid chyme now passes through the pyloric sphincter and then gets pushed into the intestines. Okay, so you're getting this and you're getting these squirts of acid chyme into the duodenum, filling the duodenum. Think of the acid chyme to have the consistency of warm porridge, right? So you're squirting this warm porridge liquid into your duodenum. Now, of course, because it has weight, it stretches the walls of the duodenum, which activates the duodenum, right? And the acidity of the chyme also activates the duodenum. And the presence of proteins in the chyme, as well as lipids in the chyme, will also activate the duodenum. And this will cause the release of gastrin. Okay? So moving chyme into the intestines causes release of gastrin. Gastrin, of course, feeds back onto the stomach's activities. Okay? So that is why the intestinal phase is a part of gastric control, because it does cause hormones to be released that do affect what the stomach is doing. Okay. All right. In a bit more detail, it mentions here in the cephalic phase, which is controlled by the medulla, which lasts about a minute. You see here you have the vagus nerve and the submucosal complex coming into play. You have postganglionic parasympathetic fibers, which can also innervate the mucous cells and chief cells and parietal cells and G cells. Gastric juice production accelerates 500 mils per hour, and it is based on your emotional state. All right, what's going on here? Remember that control of your gastric activity is usually increased by your parasympathetic and slowed by your sympathetic system, right? So when you want to activate your cephalic phase, it is going to be done via the PNS. So it mentions that the vagus nerve innervates the submucosal plexus. Obviously, the submucosal plexus is enteric nervous system um, in the submucosa. Okay, so that now will be activated, it will be triggered, which can lead to the increase in production of secretions from mucous cells, chief cells, G cells, etc. Right? And of course, if you want to slow the process down, you activate the parasympathetic nerves, which will innervate the mucous cells and chief cells to deactivate them. Okay? So once the cephalic phase starts, vagus nerve activates the submucosal plexus, your chief cells start ramping up, your parietal cells start ramping up. You make more gastric juice, waiting for food to come in, right? Phase is affected by emotional state. What does this mean? It means if you're very excited, let's say you're walking past a bakery and a gunman jump out and pull him gun on you. You can smell the lovely food in the bakery, but you're not worried about it. So under normal circumstances, smelling the lovely baked goods will cause the activation of cephalic phase. It will cause an increase in the production of mouth water. It will cause an increase in the production of gastric juice. But if you think you're about to get shot, that's the last thing that's on your mind. So your emotional state will directly, in this case here, inhibit cephalic phase. Okay? Also depression. For a lot of people, when they get depressed, they lose interest in food. So this depressed state can also suppress your cephalic phase activation, right? Now, from there, there's this lovely diagram here that summarizes what we just spoke about. You can see the vagus nerve here going all the way down in the submucosal plexus, and in the plexus activating the mucous cell, chief cell, parietal cells, all of which, of course, result in increased gastric activity, right? Now, gastric phase. Remember, this is now food getting to the stomach, and the weight of the food stretches the stomach out, and that now activates the phase. So, in addition to that, increased pH can activate the phase. So, I mean, let's say instead of eating some solid food, you drank some soup. The soup is, of course, mostly liquid, and the weight, and yes, the weight of the liquid will also impact activation. But the fact that the liquid will mix with the gastric juice and raise the pH of the gastric juice will also, of course, trigger the gastric phase. And proteins, the presence of proteins in the stomach also wakes up the gastric phase. Okay? Now, generally speaking, the gastric phase lasts 3 to 4 hours, roughly, right? Now, we have stretch receptors and chemoreceptors that have to be stimulated for the gastric phase to be activated. Remember now, the weight of the food distends the stomach, it stretches the stomach. So that, of course, is stretch receptors. Picking up the presence of proteins in the stomach. That, of course, is your chemoreceptors picking that up, right? And of course, your enteric nervous system getting triggered and increasing activity of the myenteric plexus, all of which will feed into the gastric phase activation, right? It even mentions here that you have postganglion fibers that will actually leave submucosal plexus and activate the release of acetylcholine, which will actually stimulate the chief cells and the parietal cells themselves. Okay, so all of these things are ramping up activities within the stomach. And of course, the myenteric plexus, which as we know is between the circular muscle, longitudinal muscle, this will trigger the activity of these muscles. And remember, in the stomach, you also have the oblique muscle. And these muscles will start activating rhythmically, churning the stomach, mixing the stomach content now with the gastric juice. Okay? And by the way, this reflex is one of the reasons why starving people eat dirt. I don't know if any of you have ever heard of mud cookies. Anybody ever heard of mud cookies from Haiti? It's practically a national dish at this point. Mud cookies are literally what they sound like. They're cookies made from baked mud. Okay. Um, Apparently they do it in Africa too. Haiti, of course, has their special recipe. And if I'm honest, when you look at it, it doesn't look that bad. So here we have. So they'll it's basically clay and some of the minerals mixed in. What they'll do is that they'll take these and put them in the sun to dry, right? And then once they're dry, they start to look like this, right? Now, when you eat these things, the clay is reasonably inert. It says there's no real nutritional value to it. But the weight of it distends the stomach and activate and helps to activate the gastric phase, right? And during the course of the activation of the gastric phase, it convinces your stomach that you've actually eaten food, right? And because of that, your hunger pangs will disappear. So you'll have people who are starving and they feel hunger pangs and they feel like their stomach is about to tear out of their body, as we say. The mud cookies, the non-food item, tricks your stomach into thinking it's food. And remember, just like food, it can constrict and contract it and crush it up and mix it with gastric juice just like it was food. So for a short period of time, you're relieved of the feeling of extreme hunger by tricking your stomach into activating the gastric phase, right? Now, hormones, now with regards to the gastric phase. It mentions here you have neural stimulation and the presence of peptide and amino acids that stimulate gastrin secretion. We already know that we have chemoreceptors in the stomach wall that pick up the presence of proteins in the food, right? So of course, no surprise. But this now leads to the release of gastrin. And gastrin, of course, increases the rate of activity of parietal cells and chief cells. So you get more hydrochloric acid, more pepsinogen. So the more protein you have in the diet, the more of this stuff you get. When you activate the gastric, is it the same as say opening your appetite? Opening your appetite will be more like the cephalic phase, right? So generally speaking, you don't have to eat to open your appetite. Your appetite opens, then you feel like you want to eat. So seeing food, smelling food, interacting with food, tasting food is what triggers your appetite. So you'll go from say, boy, I'm not very hungry, and you taste something somebody else is making, and all of a sudden, boy, you eat it all. So that is more cephalic phase. But obviously, cephalic and gastric phases are going to be connected because once you start eating the food, the gastric phase fully is going to trigger and it's going to make you put more and more food in, right? All right, now, where are we now? Yeah, so the gastrin, as we know, will accelerate the release of hydrochloric acid from the parietal cells and pepsinogen from the chief cells. And of course, gastrin also causes your muscles, in the muscularis externa, to become more active as well to churn the food with these various gastric juice substances, right? It even mentions here that the local distension of the gastric wall can stimulate the release of histamine, right? Now we know histamine can also trigger the activity of parietal cells, which cause, of course, the parietal cells to squeeze out more more hydrochloric acid, right? So this lovely diagram here summarizes nicely all of what I just went through. Okay. So of course, this is another important one. Then now the last phase, which is the intestinal phase. So your intestinal phase now, remember, it is the intestine causing feedback onto the stomach. Okay. So you have to have chyme. Remember at this stage here, it is going to be acid chyme. Because at this stage here, your cornmeal porridge looking mixture is going to be very acidic, right? So it gets squeezed out by peristaltic waves, pushing this acid chyme from the pyloric region into the duodenum, right? So it mentions here that you have stretch receptors in the duodenum that will pick up the weight of the food that goes in, and you have chemoreceptors there that will also pick up the presence of proteins in the food as well, right? All right. Now, once that happens, you can also have your enterogastric reflex, and the enterogastric reflex, so, is going to inhibit local stimulation of gastrin production and gastric contractions. So, and stimulates contractions of the pyloric sphincter. So, what are we saying here? When you have passed chyme into your duodenum, okay, the weight of it is there. Now, in order for you to process the food, the chyme that you've just pushed in to the duodenum, it takes a little bit of time. Nothing is instantaneous. So what you need to do is stop more chyme from being pushed into the area while you're working on the chyme that is there already. So what will happen is the weight of the food of the chyme and the presence of proteins in the chyme will trigger this enterogastric reflex. An enterogastric reflex, because it inhibits the stimulation of gastrin, less gastrin means less hydrochloric acid being produced, less pepsinogen being produced. So you're slowing down that your chemical digestion in the stomach. But it also inhibits the contraction of your stomach muscles. So you're also slowing down the mechanical processing in your stomach. So you're slowing everything down. It also, as it mentions here, causes the pyloric sphincter to contract and stay contracted. So you're shutting the door so that no more chyme can come through until you've dealt with the chyme that is there already. Okay? Now, of course, once the chyme is digested and starts moving further down your digestive system, the stretch receptors pick up the fact that there's nothing in the duodenum anymore. The chemoreceptors pick up the fact that there's no longer any proteins to be sensed, and they, of course, will relax their inhibiting of gastrin release. They will relax their forcing of the pyloric sphincter closed, so more chyme can squirt in, and the whole process starts again, right? Now, there are some other hormones that we need to talk about. We mentioned gastrin and somatostatin already. Cholecystokinin, CCP. CCK, sorry. Don't know why I was thinking about China. CCK. Cholecystokinin, as it mentions, they're secreted when lipids, in particular, and carbohydrates enter the duodenum. Okay? So we have one for proteins already, right? So gastrin. Now we have CCK. Now, CCK, as it mentions here, inhibits acid and pepsin secretion, right? Then we have, now why are we inhibiting acid and pepsin secretion? Remember what the acid and the pepsin are for. They're for breaking down proteins. If you're eating food that is relatively low in proteins, but high in lipids slash fats, same thing. Well, lipids are fats. And carbohydrates, you do not need to be processing proteins right now. So inhibiting the release of pepsin is important. And since you don't need all that pepsin anymore, you certainly don't need all that hydrochloric acid. Therefore, you produce less, right? All right. So overall, when you eat food that is not non-protein, you get less acid in the stomach. Okay? All right. So that's what CCK does. It senses that the food you're eating is low in protein, high in lipids and carbohydrates, and then stops the things associated with protein digestion, right? In it, now we also have this one here, gastric inhibitory peptide, or GIP. From this inhibitory here, I'm sure you know what this is for. As I mentioned, secreted when lipids and carbohydrates enter the duodenum. So GIP targets the pancreas and inhibits gastric secretion and reduces the rate of forced gastric contraction. Now, remember a moment ago when I said to you that the enterogastric reflex, once it senses the protein, slows down the activities in the stomach, so more chyme doesn't get pushed in while the duodenum is trying to digest this high protein high protein chyme. Well, this reflex now is for the food that is high in lipids. So the GIP now, once again, slows down contractions in the stomach, right? It slows down gastric secretions in the stomach. This, of course, means less chyme is being produced and passed into the duodenum. This once again gives the duodenum time to break down your carbohydrates so your polysaccharides to simple disaccharides or simpler polysaccharides as well. and break down your fatty acids and your lipids to fatty acid and glycerol, right? So that's what the GIP is for. Then now we have secretin.
Yes, sir.
Yes,
Second, is that why they say when you eat like fast food, you feel fuller for longer?
fast food.
Well, food with filled with fats.
Oh, fatty food.
Yeah.
Yeah, okay. I mean, honestly, this is something that we've known for hundreds of years. Y'all ever heard of the full English breakfast? If you've ever seen a full English, it's just loaded in fats and proteins. The thing is, it takes longer for your body to digest fats and protein, right? Simple carbohydrates go straight through you. So the soda in fast food is basically sugar and water. You digest that in no time. But if you're consuming like proper food, I don't necessarily mean fast food. But all right, let's say you go to KFC. Fine. Jamaican people love a KFC. You order yourself a three-piece, right? You eat the chicken, this and with the skin and everything, because let's be honest, when you go KFC, 90% of the time is the skin you really go there for. I mean, my youngest used to, when we buy KFC, he used to peel off the skin and leave the rest of the meat. That's what people really want, right? That meat, that skin is going to be high in proteins, high in fats, right? So when you consume that, because it takes longer to break down, it takes more processing to break down the proteins, it takes more processing to break down the fats. It means it takes longer for your stomach to empty. And that is why you feel full for longer. Okay? Now, if you decided instead of getting a chicken, getting some chicken, you decided, you know what I want instead? I want a chicken sandwich. Okay? Remember, it's the same KFC we're at. What we do now is we change in the order. The first of all, the quote unquote patty, chicken patty inside the burger is like a three-quarter piece of chicken that flatten out, right? So it is actually not a lot of protein, but the buns are very high in carbohydrates. So what you find is that, and there's lots of sugar and simple carbohydrates in the bun. I don't know if you noticed, but those hamburger buns are actually classified in Europe as cake, not buns. So you wouldn't consider them to be bread in Europe. You'd consider it to be pastry. That's the amount of sugar you find in them. So what happens is these very simple sugar and this hydrogenated flour and all sorts of other things actually breaks down very rapidly. So even though it looks big when you buy it, it processes very quickly. And in no time, because it takes so much less time to process this, you're hungry again, right? So if you know you have a long day, you need to keep yourself full, stay away from light carbohydrates, do consume food that is heavy in protein and lipids, fats, complex fats. That's how you stay full for longer, right? And of course, um, from strictly from the perspective of calories, it therefore means if you take the same amount of calories of cornflakes and you consume that same amount of calories of, let's say, sausage and eggs, you definitely do feel full for much longer, even though it's the same amount of calories you're getting, right? All right, let's move on. So it mentions you're secreting now. Once the pH drops below 4.5, then the secretin is going to inhibit parietal cell and chief cell activity. Now remember, this is now in your small intestine. 4.5 is low for the small intestine. Remember, the small intestine is where you're going to have digestion beginning with your pancreatic juice. Pancreatic juice, we're looking at a pH of about 7.5, 8. That's what it prefers. So when you get down to 4.5, that's very acidic. So if the small intestine is too acidic, you release secretin. And secretin, of course, inhibits parietal cells and chief cells, which of course, no parietal cell activity means no hydrochloric acid. If you have no hydrochloric acid, there's no reason to release the pepsin, right? Pepsinogen. And it stimulates the production of bicarbonate ions by the pancreas and release of bile by the liver. Of course, as you know, bile is very alkaline and it is going to, of course, help to bring the pH in the duodenum up so that your enzymes can function more effectively, right? This lovely diagram here, of course, summarizes the intestinal phase, okay? So once again, even if you don't read the rest of it, try and make sure you go through the lovely diagrams for each of the phases, since it gives you pretty much all the information just nicely summarized, right? All right, so I just mentioned this, so don't get interested again. Now, see if this makes sense to you. Protein breakdown in the presence of pepsin to form polypeptides. That sounds like C-sec business, right? Carbohydrates and lipid digestion continue until the pH through the material of the stomach falls below 4.5. So only substances absorbed by the stomach are fat soluble, including alcohol and aspirin. The stomach lacks an absorptive surface, unlike what the small intestine has. Okay? So I'm hoping you've seen these points before and it makes sense to you. Now, by the way, this is one of the reasons why you can get drunk very quickly when you drink strong alcohol, which of course is point 3. Because alcohol can pass easily through your stomach wall, um, drinking the same amount of alcohol in beer versus vodka, the vodka will put you on your backside a lot faster, right? Because when you drink alcohol that is very high, when you drink drinks with very high alcohol contents, diffusion gradient, remember? So the alcohol diffuses more rapidly from these drinks in your stomach into the blood vessels, capillaries running along the stomach. And then from there, of course, the alcohol can run to your liver and then to your brain. And then before you know it, you're drunk, right? So you've ever seen people do these silly challenges where they give them like half a bottle of vodka and say, drink it. And they'll drink half a bottle of vodka and a minute later they'll be like, hey, they're still good. 5 minutes later, they're still good. By the time you reach 10 minutes and pass out, that's what's going on, right? The thing with vodka is because the taste of the vodka is not particularly unpleasant, unlike, say, gin and some other things. And it's easy to mix vodka into everything. You'll be drinking some drinks that are very high, 40% alcohol content, and you won't even know. And so you've had like 2, 3 of them because they're full of sugar as well. So they're very sweet. And before you know it, you're on your ass. Anyway, now, small intestine. 3 main parts, right? Duodenum, jejunum, ileum. We're looking at our section to the jejunum this week. Um, as it says here, 90% of absorption will occur in the small intestine, right? I'm sure you've all seen a diagram like this before, right? So major parts, if you notice, the duodenum is by far the smallest piece. It's just this little piece here. So from the stomach to the curve around the pancreas, that's basically all the duodenum is. Okay? And then of course from there we have our jejunum. Okay? And then the last part is our ileum, as I said before, right? Now, the purpose of the small intestine is, as it mentioned before, for chemical digestion mainly and absorption. Okay? And of course, the pancreas is extremely important in the process of chemical digestion, as I'm sure everybody knows. Now, you should already have met these terms at CSEC. So pancreatic amylase, as we know, amylase breaks down starches, complex carbohydrates. Then we have pancreatic lipase. Of course, a lipase breaks down a lipid. And of course, nucleases, which will break down your nuclear acid. So we're talking about DNA, RNA, right? And of course, we have our proteolytic enzymes that complete the digestion of proteins, right? All of these things are coming from the pancreas. Now, of course, the liver and the gallbladder, the gallbladder really is what the liver is providing, right? The liver makes bile, passes the bile to the gallbladder here, and in the gallbladder, what happens is that you absorb water from the bile, so you're concentrating the bile to get it nice and strong, okay? And then now, what happens is when you have a high concentration of fats in your duodenum, then CCK is released, and it causes the gallbladder to start contracting, and that pushes bile down the cystic duct to the common bile duct, all the way down until the common bile duct reaches right here. So this area here called the hepatopancreatic ampulla. And from there, the bile goes right into the duodenum. Okay? So where it can mix with the very fatty food you've eaten. And remember, bile is not an enzyme. Bile is an emulsifying agent. It is the job of bile to take the fat droplets, big fat droplets, and make small fat droplets. And that's it. Okay? Now, why is it doing that? Because the lipase enzyme that we make as human beings is not good stuff. It's mediocre at best. So you need as much of a large surface area as possible to speed up its efficiency. Hence, you need the emulsifying properties of bile. Okay. Now, while that is happening, of course, your pancreas is not idle. Your pancreas will pick up now the presence of proteins, for example, in food, and carbohydrates, and your pancreas will release pancreatic enzymes through the pancreatic duct. The pancreatic duct now will also merge with the hepatopancreatic ampulla and dump your pancreatic juice with all those lovely enzymes into your duodenum. Okay? So now you have bile in there, you have pancreatic juice in there, you have your chyme in there, everybody mixing up together now. And digestion, of course, chemical digestion can really get going, right? So this one here just shows you how the ampulla in all its glory. So you have here's your your common bile duct here. Here's your pancreatic duct, and they merge together right here. Okay, before everything enters the duodenum. Now, this is actually a bad system. Okay, the evolution was very, very lazy with this one. Evolution really should have put two separate tubes. Common bile duct should have attached separately from the pancreatic duct. Here's the reason why. You get a gallstone. Gallstones are very common. Okay, the gallstone can actually travel down your common bile duct and end up getting stuck. Sometimes it's just temporary, right? But even if it's stuck for a couple of days, right here in your hepatopancreatic ampulla, this gallstone now is preventing the release of bile into the duodenum. It is also preventing the release of digestive enzymes from the pancreas also into the duodenum. This now means because of this gallstone, you're eating food, you're chewing the food, you're swallowing the food, food is passing out. You're not getting very much out of the food because you're not able to properly digest it, right? And you can, technically speaking, die slowly of starvation this way because you're eating and eating and eating, you just can't digest the food, right? Unless, of course, as the pancreatic juice builds up now in the pancreas itself, it puts pressure on the pancreas, and next thing you know, you have pancreatitis. And that in itself can lead to the pancreas rupturing, and you can die. So yeah, it's not a great system, right? Not a great system. Anyway, let's talk about the general structure of the small intestine, right? Now, plica circularis. I'm assuming at least some of you have heard of plica circularis, right? The plica circularis is going to be involved in increasing the surface area of your small intestine, and it is also involved in something called segmentation I mentioned to you before. Okay? Um, let's use this one. So the plica circularis refer to these folds here in your small intestine. Okay? You can start to see them here so as well. So these folds you see in your small intestine, um, they do increase surface area, as I mentioned a moment ago, right? But because they're of the circular longitudinal muscle, it also allows you to do this thing where you can push the food forward and push the food back with the folds helping. Which of course, if you're moving the food back and forth, mixing it with digestive enzymes, that is segmentation we're doing, right? So the plica circularis increases surface area of the small intestine. So you have more surface area for secretion and absorption. And it also aids in segmentation, right? Villi, of course, we all know villi. These are little villi you see coming up here from the wall of the basically coming up from the mucosa. Of course, the jejunum is loaded with these villi. You'll be drawing some of them. And this, of course, increases surface area for sure. Then now you also have the microvilli, which come off of the villi, which are also projections, and they further increase surface area. So plica circularis, villi, and microvilli, all of them lead to an increase in the surface area, the small intestine, which means, of course, more absorption, faster absorption, right? So they're very important. Now, I have not mentioned lamina propria yet, but of course, for the Monday class, you already know what it is. Lamina propria is this layer of connective tissue you find right below your epithelial layer. As it mentions here, it has capillaries that can do gas transport, absorb nutrients as well to the hepatic portal vein. You will find some components of your lymphatic system in there. So your lacteals, which remember are lymphatic capillaries, will transport very large substances or fats to the blood via the lymphatic system. Okay? And that will be your chylomicrons. And we'll talk about chylomicrons in a bit. Then now the muscularis mucosa, as we know, is a muscular layer that is the lowest part of your mucosa, and it helps with moving the villi. Right? It squeezes the lacteals, which helps to move substances through them on their way to the lymphatic vessels as well, right? Obviously, this is a lovely diagram, and it would be nice if you learn what's in on this one, right? So of course, this is another lovely diagram, but this one actually shows you more clearly, I should think. Here's your mucosa. Okay, here are my lovely villi. And when you look at your lovely villi, you can see the cells here, these columnar cells, have little brush border, which of course are the microvilli, which aid in absorption. And of course, within the villi, you of course have your lymphatic capillary, which is your lacteal. Remember, of course, lacteals are for the absorption of large molecules that can't pass easily, and also for fats, fatty acid and glycerol. Why? Remember that fat-based substances will not dissolve directly in the blood because, of course, hydrophobic, right? So you pass them through the lymphatic system first, where you can attach them to carrier proteins before you send them back to the blood. All right, so we're looking at the three main parts now, right? All of the small intestine. Let's see how many of our major sections that they have each. So Your duodenum, for example, does not have a lot of plica circularis. Okay? You're not doing a lot of absorption in the duodenum. Mainly in the duodenum, you're doing, of course, is chemical digestion. You'll notice it has numerous villi as well as mucus producing glands though. Okay. Then now you have your jejunum, which has lots of plica circularis, and it also has the most villi and microvilli. It mentions it's prominent in its proximal portion. So that basically means that the section of the jejunum closest to the duodenum is loaded with villi, loaded with plica circularis, because you're doing a lot of absorption and digestion here. Okay? Then now the ileum, which is the furthermost part, has very few villi, which automatically tells you you're not doing a lot of absorption here. There's no plica circularis at all, and there are no pairs, there are no pairs patches, which of course are lymphoid tissue near the end of it. Okay? So each section is designed to do something, different things well. Okay. So duodenum does digestion very well, and it does some absorption as well. Jejunum does absorption very well, right? The ileum doesn't do digestion that well, but it does absorb some water and some other substances as well. It's not really for that specifically. The jejunum is by far the most important when it comes to absorption, right? Particularly the section near the duodenum. That said, let's talk about some of the substances that you'll find. Obviously mucus, because mucus is absolutely everywhere. We also have brush border enzymes. So enzymes secreted by the brush border cells, right? We have enterokinase, maltase, sucrase, lactase, dipeptidases, and peptidases. Now I'm hoping you would have recognized some of these terms, right? But I'm going to give you a moment to think about it because I have a table or two that will give you the breakdown of what these enzymes, these groups of enzymes will do. But think about it now. Enterokinase, maltase, sucrase, lactase, and dipeptidase and peptidase. What are these things doing? All right. Movement through the intestines. Obviously, we're talking about peristalsis, talking about segmentation, as we mentioned before, right? And we've gone through this already, so we don't want to spend too much time on it. But peristalsis, segmentation is what we're talking about. Now, just to refresh you, peristalsis, remember, you're moving substances one way, one direction only through your intestines, right? So it mentions your ring-like peristaltic contractions sweep food along the GI tract. Basically, the GI tract behind the food, the bolus contracts, squeezing the food, pushing it forward. Then when the food gets pushed forward, the section down here so relaxes, and in the section near the front again, squeeze near this part will squeeze. So you're doing incremental squeezing from the back to push the food forward, right? Remember in segmentation, you're moving in back and forth direction, right? So you have muscle contractions breaking chyme into several small pieces, and until the chyme is thoroughly mixed with the digestive juices. So here now you're moving the chyme back and forth, right? You're breaking it up so you can and mixing it with the intestinal juice, pancreatic juice, so that you can promote the process of chemical digestion, right? Now, it mentions here that your two reflexes coordinate activities along the entire length of your small intestine, right? You have the gastroenteric reflex, which causes an increase in motility, and it also causes an increase in secretion along small intestine, right? So you're pushing the bolus along with the gastroenteric reflex, and as you're pushing that bolus along, you're also increasing secretion of substances onto the bolus itself. right? Then we have the gastroileal reflex, which causes the relaxation of the ileocecal valve, and that is increased by gastrin secretion. All right, the ileocecal valve. Okay, the nice thing about these valves is that the name tell you what's going on with them, right? So an ileocecal valve is going to be between the ileum and the cecum, right? Of course, cecum, as you know, is going to be our large intestine, okay? So the ileocecal valve is going to be that is going to allow substances to leave the small intestine and enter the large intestine, right? So the idea here now is that once you've absorbed the bulk of whatever you want to absorb from your dinner, you want to now push what is mostly waste into the large intestine. So you relax the ileocecal valve and allow, of course, for the pushing of the substances into the large intestine out of the small intestine, right? Now, why would gastrin want this to happen? Remember, gastrin is increasing the activity of your stomach, right? Including the production of chyme, which leads, of course, to acid chyme being pushed into the duodenum, the small intestine. So if you're emptying the small intestine on one end and refilling on the other end, you have a nice continuous process. Okay? So the same enzyme that speeds up the filling at one end helps to speed up the emptying at the other end. Right? That's why this enzyme is involved in both. Okay? Now, what is the pancreas releasing? We all know about pancreatic juice. Okay? There are lots of different things in the pancreatic juice. But one of the main ones, of course, is bicarbonate fluids in the juice, which help to neutralize the acidic environment that the chyme will produce. Okay. Mentions that you have endocrine cells of the pancreatic islets, which will produce insulin and glucagon. Now remember, endocrine goes into the blood, exocrine goes into spaces or onto surfaces. Okay. So the alpha cells that produce glucagon and the beta cells that produce insulin are parts of the endocrine system. Okay. In the exocrine section, now we have the acinar cells, and they empty their products into epithelial ducts, which will produce the pancreatic juice. Okay. So they're emptying into a duct. Duct is carrying the substances into the stomach, secreted onto the inner surface of the sorry, my stomach, onto the inner surface of the intestines. Therefore, these are exocrine. Okay, we already went through this diagram already. We're not going to do it again. So in the pancreatic juice, we mentioned already the bicarbonates. So sodium bicarbonate, which acts as a buffer solution. So it helps to raise the pH above 4.5 up to 7.5 to 8.8, right? Then, of course, pancreatic enzymes now. We have cholecystokinin, second time I've seen this enzyme, this hormone, will actually accelerate the production and secretion of pancreatic enzymes and the relaxation of the hypopancreatic sphincter and the contraction of the gallbladder, right? So why is it doing this? Remember, the CCK is responding to the high concentrations of lipids and carbohydrates, right? To digest lipids and carbohydrates properly, efficiently, you're going to need bile from the gallbladder. So you, of course, will increase the contraction of the gallbladder while you relax the ampulla and the hypopancreatic sphincter. You're also going to need the enzyme lipase, which of course is coming from the pancreas. So Once again, relaxing the hypopancreatic sphincter ampulla will allow the lipase to come in. But remember, the enzyme also increases the production of these enzymes, right? So cholecystokinin increases the production of lipase, increases the production of your carbohydrates, all of which will then be present for release onto the food that is high in carbohydrates, high in fats, right? So you're getting the bile because of the CCK. You're getting the pancreatic juice with the lipase and the carbohydrates is because of your CCK. All of these things converge together in your duodenum so you can digest protein, so you can digest carbohydrates and lipids properly, right? So of course, pancreatic amylase, no surprise, it's just like regular amylase. It takes your carbohydrates, your complex carbohydrates like starch or amylose, breaks them down to give you, of course, simple carbohydrates, disaccharides, and short up oligosaccharides, if you know what those are, right? Oligo now just means short, right? Then now we have pancreatic lipase. We already know what lipases do, right? And we already know what nucleases do as well. So what else do we have going on there? Our proteolytic enzymes. Now, some of these you probably know already, okay? I'm hoping that some of them you know already. But enterokinase, this one is very important. You may not have heard of this one before, but you need enterokinase to activate trypsin. Trypsin is one you should have heard of from CSEC, right? So you need it now to turn on your trypsin. Once you activate trypsin, trypsin can go on to activate chymotrypsin, and this can activate carboxypeptidase and elastase, okay? Which of course will digest your proteins and which gives you your dipeptides, tripeptides, and finally the amino acids. So in order for you to properly digest proteins, remember now, you need enterokinase first because enterokinase will activate your trypsin. Then trypsin will activate the other enzymes associated with protein digestion. That is why enterokinase is so important. Now, this lovely diagram here compares two other types of protein digesting enzymes. You have endopeptidases and exopeptidases. Obviously, endo means inside, exo means outside, okay? But let me nutshell this thing for you. Here's the basic idea. Let us say we have, let's say this is a polypeptide chain we have here, right? Like this. An endopeptidase's job is to go within the chain and target the bonds between amino acids, okay? So we'll then take this amino acid here and break it down. Say here amino acid, this polypeptide here and break it down. So now you end up with several dipeptides. Let's assume that's correct, like this, okay? So you're taking a longer chain, breaking it into shorter chains. Now, since you have three pieces, three separate enzymes can attack these three pieces at the same time, breaking things down quickly, okay? The exopeptidase works a little differently. So if you have our chain again, exopeptidases actually target the amino acid bonds near the ends. Now, this means, of course, when the exopeptidase is done, end up with individual amino acids. right? The endopeptidases will give you dipeptides. The exopeptidases will give you amino acids. So you're going from a dimer to a monomer. Okay? So that's why you need both. You need the endo ones to chop the big chains down into small pieces, and then you need the exo to chop the small pieces down into individual amino acids, right? So it mentions here that pepsin, trypsin, and chymotrypsin are endopeptidases. Okay? They take big, long, complex proteins and break them down into smaller chunks of polypeptides. And then carboxypeptidase and aminopeptidase now, of course, are exopeptidases, which take your chunks and cut them into individual amino acids, right? Now, this lovely diagram here summarizes the functions of the various enzymes that we've mentioned, where they're released, etc., etc. This is a lovely table. I would advise that you have a look at it at some point, right? All right, it's 6:04. I'm going to need another 25 minutes or so to finish up, so please bear with me. All right, liver. Obviously, with regards to digestion, the liver's main contribution is going to be bile, right? We've mentioned bile several times before. We know bile is there to emulsify fat, right? It also mentions here that the liver has many, many other functions. As a matter of fact, it has more functions than any other organ in the body. It is very busy, right? So metabolic regulation mentions carbohydrate regulation, lipid, amino acid metabolism is all involved in that. Waste production and removal, vitamin and mineral storage, drug inactivation. Liver is doing all of these things, right? The liver even helps with blood regulation. So very busy. Of course, for our purposes, bile is what we really want for this stage, right? So what is bile really? Bile contains some amount of water, right? As well as various electrolyte ions. It contains dilute and buffered acids substances. It contains bilirubin, contains cholesterol and bile salts, right? All of these things, of course, enhance function of bile, which is the emulsification function, right? But of course, the buffers here will help to control the acidity of the chyme as well, right? So the pancreas is trying to control acidity, and the bile helps as well. Now, of course, bile is released, as it mentions here. Bile is secreted from the liver in the presence of secretin when lipids enter the duodenum. It also mentions that bile will emulsify fats, as we know, and facilitate the absorption of lipids by the intestinal epithelium. All right. So overall, bile helps to break the fat down, and it helps the fatty acid and glycerol to be absorbed into the lining of your digestive tract, right? And we already know bile is stored in the gallbladder, etc. And I already mentioned to you that the gallbladder, of course, once you make the bile and you pass it to the bile isn't ready yet. Okay? It is still too watery. So as the bile sits in the gallbladder, you absorb water from the bile and you make it more concentrated. And of course, bile is also triggered by CCK. And we already went through what CCK does, how CCK increases contraction of the gallbladder wall, how it increases the contraction of the common bile duct, how it causes the relaxation of the hepatopancreatic sphincter, and all of these things, right? Which of course lead to bile being secreted into your duodenum, but hopefully onto your food high in carbohydrates and fats, right? I mean, the number of times you've seen this diagram, it should be fairly obvious it's important, right? Now, I mentioned gallstones earlier. So here are a few choice ones that have been extracted from somebody's gallbladder, right? They're generally formed in areas of very high cholesterol and high oxalate concentration as well. Okay. So if there's if you are constantly consuming food that is very high in cholesterol, it increases your risk of gallstones. Yes, you do need cholesterol to make to make the bile in the first place. But as we always say, too much of anything is not good, right? But you also need to watch oxalates. Oxalates are found in nuts. Oxalates increase the formation rate of gallstones by acting as nuclei for the formation of the stones, right? So if you're somebody who likes peanuts and cashew and all of those things, and you eat them regularly, you also increase your risk of getting gallstones, right? But only by a little bit, unless you do not drink enough water. If you drink a lot of water and you eat all of these lovely peanuts and cashews and everything else, it has virtually no impact on your chance of forming a stone. But if you're one of these people who drink your tea in the morning and then the next time you see any liquid is afternoon, and then during the course of the day you eat some peanuts and whatever, and you do that regularly, you dramatically increase your risk of getting gallstones, right? Because the idea here, of course, is that the more you drink, the more water ends up in the bile that you're producing. So it takes longer to pull the water from the bile in the gallbladder, right? And of course, by then, you'd have eaten something resulting in a release of the bile before the bile gets so concentrated, you start to form stones. Okay? So yeah, everybody says it, and it's not wrong. Drink plenty of water. It's not even a joke. Drink plenty of water. If your body doesn't want it, it'll pee it out. Just you make sure you're drinking plenty of it, right? And soda, I suppose soda counts as water. I know these days that some people drink only soda. But try to avoid soda that is very high in sugar, please, because that also can slightly increase your risk of getting gallstones as well. All right. All right. Brush border enzymes. We mentioned them earlier. Maltase, sucrase, lactase, dipeptidase, and peptidases. Now, maltase, of course, breaks down maltose, or malt sugar, as you know from CSEC. Sucrase breaks down sucrose, which is cane sugar, as I'm sure you know. And you get glucose and fructose when you break down sucrase. And of course, you get glucose when you break down maltose. And I'm sure everybody remembers that glucose and fructose are isomers, right? Then lactase breaks down lactose. And of course, lactose is where you get lactose intolerance from. When you break down lactose, you get glucose and galactose, which are also two simple sugars. Now, lactase production is something that is tied in to your environment and to genetics. Certain sub populations of humans are less likely to become lactose intolerant than others, right? I prefer not to use the term race. Instead, I say subpopulation because guess why? Human beings are not genetically diverse enough to be considered different races, okay? At least not from an actual scientific perspective. So if somebody tells you a race is a social construct, it literally is a social construct. There's no biology in it, okay? So, for example, your subpopulation of Irish tend to have lower incidences of lactose intolerance than subpopulations of Japanese, right? And a part of that has to do with diet over centuries. Japanese people rarely consume significant quantities of milk into adulthood. It's not something that's in their diet, okay? Japanese people don't consume that much cheese or drink that much raw milk or any kind of milk, really. I mean, soy milk is not real milk, okay? It's Soy milk is a misnomer. It should be soy juice. Anyway, whereas Irish people spent a lot of time with cheese and milk and other dairy products for centuries, they've been consuming these things, large part of their diet. Because of that, they naturally make more lactase than somebody who is Chinese or, well, Japanese or Chinese, right? And as you get older, you make less and less lactase, particularly if you stop consuming milk-based products for a while. um, your lactase production might end completely. And once it stops, it doesn't start back up, right? So let's say you're eating your cheese and your eggs and everything is fine. You decide to go on a no cheese diet for 6 months. At the end of the 6 months, you come back and try to eat the same amount of cheese you normally do. All of a sudden, you will get the symptoms of lactose intolerance because you're no longer producing lactase enzyme, right? Whereas once again, somebody who's from an Irish descent can go much longer before this happens, right? Now, dipeptidases and peptidases, of course, break down dipeptides and tripeptides to give us our amino acids, right? Now, as I mentioned here, substances are absorbed in the small intestines are usually going to be monosaccharides, which are absorbed by the duodenum and the upper part of the jejunum. Remember, as we said too, these are the two parts most involved in major absorption. Co-transport with sodium. So that means you're absorbing sodium and at the same time, you're absorbing simple sugars. So they're moving at the same time, co-transport. Amino acids, however, are absorbed mostly at the end of the jejunum and are also co-transported with sodium. Now, I want to point out how important sodium is to absorption, right? So if you are consuming food with extremely low salt content, it is actually harder to absorb the food, right? Particularly if you're talking about carbohydrates and protein-based foods. So in a way, that's one of the reasons why the fast food blows through your system so quick, because the fast food is full of salt, right? So plenty of sodium and chloride there to act as co-transport mechanisms, right? In terms of our fats now, you're absorbing them in the duodenum and the jejunum and in the upper part of the ileum near the jejunum, right? Now, these guys, remember, cannot go directly into the blood because, of course, they are hydrophobic, right? So what happens instead is that you form what are called micelles, which are water soluble, and you then will pass your fat, you form the micelles now out of your fatty acid and glycerol. And then the micelles will pass through the epithelial cells of the villi. And once they get inside the cells, they reform our glycerides and our fatty acid. And then so here so you can see here you're forming triglycerides. And in the triglycerides now are going to combine cholesterol and lipoproteins, which is important here, and phospholipids, and that gives us our chylomicrons. So you have lipoproteins, your triglycerides, and phospholipids all come together to give you the chylomicrons. And the chylomicrons can pass through into the lacteals, and from the lacteals they can pass through the lymphatic system and eventually make their way back to the blood. Okay? So this here is showing us a simple diagram representing um, some of the breakdown and transport, right? So here we have our carboxypeptidase. And the carboxypeptidase, of course, as you can see here, you're breaking it down into amino acids, and the amino acids are absorbed directly into the blood. And the aminopeptidase breaks down to give us our amino acids, same thing, right? Dipeptidases, same thing. So you're breaking down these short chains of amino acids to give us monomers, and then the monomers can be passed right to the cell membranes via channel proteins, and from there make their way straight into the blood. They need channel proteins to come in, of course, because Um, there are light their cell membranes are hydrophobic. And of course, anything that dissolves easily in water does not pass easily through the cell membrane. So therefore, you need channels, as in the co-transport system we mentioned earlier, to help move these things from the lumen through the cell, through your intestinal cells, and eventually into your capillaries, right? So it's, as it mentions here, brush-border enzymes finish tasks by producing free amino acids that are absorbed into the intestinal epithelium, as we mentioned here, and sodium-dependent amino acid co-transporters move amino acids into epithelial cells. So remember now, co-transport, you're moving sodium, you're moving amino acids at the same time into the cell. Then from there, you move them same time again into the bloodstream via capillaries, right? Now, this here now is showing us how fats are absorbed, okay? Which once again is going to be much different. So here you have your fat globule. You have your emulsification occurring. So you're taking this one big fat molecule, breaking it down into several small molecules. And then now, once you have your small molecules, you notice, as it says here, fat droplets coated with bile salts. And the bile salts help to prevent these small fat globules from reattaching to each other. Okay? So it keeps them as separate little drops. They're like little bumpers in bumper cars, right? So they can hit each other and bounce off without reabsorbing, right? Now, as we mentioned before, you take your fatty acids and your monoglycerides, and then you're going to form your chylomicrons. Then now the chylomicrons, remember, have to all right, let's start from the top, make it easier. So I'm going to read what's here and explain as I go, right? So as I mentioned, you have fatty acids and monoglycerides associated with micelles in the lumen of the intestines. So these micelles here will contain the fatty acid and glycerol that you broke down. Okay? Then the micelles, because the micelles are lipophilic, they love fats, which means they can easily merge with your cell membrane here. Whatever they contain can be passed into the cell um, via the cell membrane because the micelle can merge with it. Okay? So that's the first part here. Then it says fatty acids and monoglycerides resulting from fat digestion leave micelles into the epithelium by diffusion. So what we just mentioned, right? And it mentioned the second part now, fatty acids are used to synthesize triglycerides in the SCR. So remember what I said to you, your monoglycerides are combined to form triglycerides. You're using your smooth endoplasmic reticulum to do that. And once you've done that, you package these triglycerides and pass them to the Golgi apparatus here. So fat globules are combined with proteins in the Golgi apparatus to form chylomicrons. And the chylomicrons will then pass to the cell membranes where they're dumped out of the cell and then can pass into the lymphatic capillaries. And from there, of course, they get transported to the blood. Okay? All right. This is summarizing digestion, so we can skip that for now. All right. Let me see, where are we now? Okay, cool. We don't have much left. We only have about 14 slides and we're done, right? Now it mentions here that vitamin C and B can be reabsorbed easily by just normal passive diffusion. B12 with intrinsic factor, okay? Then vitamins A, D, E, and K, which are fat-based vitamins, can be moved in via micelles. Remember, micelles form around the vitamins and then pass them into the intestinal cells, right? Because the micelles are, of course, lipophilic. This lovely diagram here, it does a breakdown of everything we've just said about digestion and absorption. So I'm going to suggest that you go through this one. Okay. All right. So we leave the small intestine and now we're in the large intestine. Last part, right? Now, the first part of it here is the cecum. It's the blind ending part. Then from the cecum, we get the colon. The first part of the colon is called the ascending colon because, of course, you're moving the food up. Well, let's call it feces at this stage. You're moving the feces up. Then move the feces right to left. So, of course, that's transverse. Then you move the feces down, so that's descending. And then the last part of the colon bends to go down to the rectum. So we call that the sigmoid colon because it bends in sort of an S shape. Okay? And that's where you get the names of these parts. Right? The rectum, of course, is a modified section of the colon that's modified for storage of temporary storage of feces. And no surprise, the anal canal leads to the outside. Right? Nice C-sec explanations for this stuff, right? Now, your large intestine, simple column epithelium again, right? But of course, remember what I said about the rectum, stratified squamous. Now, no surprise, the large intestine has lots of mucus secreting cells. Because, of course, as you're moving the waste through the large intestine, you're continually absorbing water from that waste. So as you're moving along, the waste is actually getting denser and denser and more organized, right? Now, one of the problems, of course, is that if you don't drink a lot of water, you will find that you quickly pull water out of the feces, and the feces becomes very hard, and it makes it harder to move. So next thing you know, you have constipation, right? So believe it or not, if somebody suffers from constipation, spending several days drinking lots of water actually will help with constipation because it will soften the feces over time, right? Anyway, so as it mentions here, the mucus being produced so to protect the intestinal wall and it helps to hold the fecal matter together and it even contains sodium bicarbonates that are going to help to neutralize acids. Where the acid is coming from, your large intestine is full of various types of bacteria. Now, that is nothing to be alarmed about because these bacteria have evolved to live in you. They are part of your bacterial fauna. Okay? They're supposed to be there. As a matter of fact, these bacteria do several important things for you, including help to release vitamins from even the waste that you've passed to them. So they actually provide nutrition. So they're useful stuff, right? You don't want to live without them. So neutralizing the acids they produce, it's a small price to pay for being able to get more vitamins from your food, right? And of course, this is what the large intestine looks like in the histological section. Now, when you look at it, it's going to look similar in a lot of ways to the stomach, right? I'm hoping you notice that. You have intestinal glands that look very, very similar to gastric pits, okay? As you can see here. And you'll also notice the mucosa is heavily inundated with capillaries, as well as lymphatic vessels, and lots of glandular tissue here as well for the production of mucus, right? You also have a muscularis here because peristalsis is still a thing. But if you notice here, the muscularis mucosa is very thin, and the muscularis externa is not particularly thick either, right? Why is that? Think of it this way. You have absorbed most of the major nutrients from the food that you've consumed. You have absorbed most of the water from that food. So by the time you get to your large intestines, the volume of substance that you're moving through is much smaller than the large intestine moved, or that sorry, than the small intestine would have moved. So you don't need as much muscle, right? Of course, in our submucosa, you're also going to have a fair amount of blood vessels. And then we have our serosa, which is, of course, our outermost layer, as we all know, right? So as it mentions here, once absorption has taken place in the ileum, undigested food is pushed into the large intestine. remember the ileocecal valve, right? And the job of the large intestine is mainly for the reabsorption of water. Because of course, water is a very important asset to us, right? So you want to pull as much water as possible as you can out of the feces before it is ingested. Because you do, no matter what you do, you're going to lose some amount of water in the feces. So you want to limit that as much as possible, right? So it mentions is the large intestine maximizes the surface area for absorption of water by being long and being folded, right? And the mucus secreted to lubricate the passage of the feces, of course, will help, and the muscular contraction, same peristalsis, helps to keep the feces moving. Now, here's the thing. You need to have fiber in your diet. The reason why you need to have the fiber in the diet is because when the feces first passes into the large intestine, it is very sticky, right? In a lot of ways, it's very similar to peanut butter, which means it's harder to push it along by peristalsis. By mixing in fiber, right? Those bits and pieces of cellulose, you have to give structure to the feces. And with structure, of course, it's easier to push the feces along. So think of it this way. Imagine, once again, you're holding a nice big chunk of peanut butter in your hand. Nice and mushy. But of course, there's not much you can do with it because it's sticky. Then, of course, imagine yourself sprinkling sawdust into that peanut butter. And the more and more sawdust is sprinkled into the peanut butter, the more it absorbs sawdust and becomes less, it becomes less sticky and is held together better. And before you know it, you have a ball of something that's more like Play-Doh than peanut butter, right? Just by adding that little sawdust, which of course would be, or roughage, right? Now, we mentioned bacteria earlier, right? So let's talk about some of the things that the bacterial fauna can do for you. So we have here, it breaks down indigestible material, which is great, and leads to the production of B complex vitamins. So you're automatically getting B vitamins from it, as well as vitamin K. So you're able to extract more B vitamins as well as vitamin K from your leftovers because of these bacteria. So they're pretty important little buggers, right? So your intestinal fauna is very important, okay? And that is one of the issues with antibiotics. When you take antibiotics, they're indiscriminate. They will wipe out your intestinal fauna, the good, the bad, and the ugly, as the saying goes. So it actually ends up people who take a lot of antibiotics having a reduced ability to absorb nutrients in the large intestines in particular, right? And in addition to that, without the bacteria being present in your large intestine, it can actually cause your immune system to malfunction. Your immune system is used to the bacteria being there. It's used to getting feedback from the bacteria there. When you remove them, you remove that feedback. And the problem with your body as a human being is that your body has a tendency to, if it doesn't hear anything, it just makes it up, right? So without input from the bacteria, your immune system can start to overreact, can actually start attacking the tissue lining your digestive tract, leading to all sorts of gastrointestinal problems, like GERD, for example. Uh, Crohn's disease, sorry, not GERD, like Crohn's disease, right? Now, this diagram here, of course, is just a very brief summary of the basics of the digestive system. So let's go through whenever you have time, right? It's pretty straightforward. So your large intestine is going to produce water and electrolytes when it is irritated. So the idea is that if some bacteria or something noxious makes its way all the way through your intestines and gets to your large intestine, it is going to trigger the increase in the production of water, which is passed into the large intestine, as well as electrolytes that are passed into the large intestine. And then it can also cause increase in peristaltic contractions. Now, I'm hoping y'all will have recognized what's going on by now. What you're doing is essentially like you're creating diarrhea. So some unknown substance goes into the large intestine. Your immune system responds by flushing the system out with extra water from your body and extra electrolytes. Now, of course, the loss of electrolytes is why you when we were talking about electrolytes last week, we mentioned diarrhea causing things like hypokalemia, for example, right? Now, movement of the feces is once again due to peristalsis, right? And segmentation can occur here as well, moving the food along, or the feces along, I should say. Now, I mentioned lactose intolerance already, so I don't think I'm going to mention it again, right? And that's it. We are totally done. Now, awesome. That now means, um, yeah, I was having problems with Moodle earlier in the week, so I wasn't able to open it. I'll check again after class to see if I can still reopen. As I said to you, I'm reopening all of them because it's been a messed up semester. So let's take things easy. All right. Anyway, so we're good where this is concerned, right? Now, what I do want to do oh no, not Friday. We can't do it on Friday. I'm putting links in Moodle on the different topic headings, right? So this one you'll find the Moodle link on the digestive system, but it's gonna take a little while before I can put it up. So don't check before tomorrow. I was hoping to do online on Friday. Unfortunately, we have lab on Friday, so we can't do that. As you know, next Tuesday we're going to have our in-course test, right? I'm told that you'll be having lots of in-course tests in the first week of December. So I don't think it's a good idea to have it then, right? Then again, I am somewhat flexible.